All right, let's dive into something that sounds like it's straight out of a sci-fi movie: growing miniature human eyes in a lab. And no, this isn't Hollywood. It's a real technology called retinal organoids, and it's giving us this unbelievably powerful new way to understand what causes blindness, and maybe, just maybe, how we can finally cure it.
To really get why this science matters so much, let's start with a really human problem. I mean, just imagine for a second that the world around you is slowly getting dimmer. The colors just aren't as bright, and the darkness of night starts creeping into your daytime. This is the reality for millions of people living with inherited diseases of the retina.
And right at the heart of our story today is one of the most common of these conditions, what you could truly call a disease of darkness. That right there, that's the classic and devastating description of a condition called retinitis pigmentosa, or RP for short. It usually starts with just having trouble seeing in low light, but over time, it can progress all the way to complete blindness.
So what's actually going wrong here on a cellular level? Well, RP is basically a group of genetic disorders that go after the retina's photoreceptors, the rods and cones that are supposed to detect light. And while lots of different gene mutations can cause it, a really common culprit is the gene that makes rhodopsin. That's the super important protein that lets our rod cells see in dim light.
So, for decades, fighting this disease was a huge challenge. I mean, to understand it, you have to be able to study it. But how in the world can you study a living human retina as it degenerates without harming a patient? Well, the answer lies in something you might call a cellular time machine.
Okay, this is where things get truly incredible. Scientists can take a simple blood sample, pull out a patient's adult cells, and basically rewind their biological clock. They use this reprogramming process to turn them back into what we call induced pluripotent stem cells, or iPSCs. You can think of them as total blank slates. They have the potential to become any kind of cell in the entire human body. This gives researchers a virtually unlimited, personalized supply of a patient's cells to study.
But that, of course, is the big question, isn't it? It's one thing to have the raw materials, but how on earth do you guide those simple blank slate cells to build something as intricate and complex as a light-sensing eye?
Well, the answer lies in an incredible biological recipe, a step-by-step guide that carefully copies the way a human eye develops naturally.
Just look at this timeline. It really shows you the sheer patience this takes. This is not a quick process. Over months and months in a dish, these stem cells go through this precise developmental dance. By day 16, you see the first little hints of an eye, these things called optic vesicles. By day 30, the tissue has organized itself into what we call an organoid, a tiny 3D miniature version of an organ. And from there, it just keeps going, with those all-important rod photoreceptors showing up around day 120. It takes more than 200 days for these mini eyes to fully mature and grow the little structures that actually sense light.
Now, this process is mind-blowing all on its own, but its true power, its real magic, is unlocked when scientists do all this with cells taken directly from a patient who has retinitis pigmentosa. This is where we get to see a sickness in sight.
So, to get to the bottom of the disease, the researchers set up this perfect side-by-side comparison. They grew one batch of these retinal organoids from a healthy family member. That's their control group. And then they grew a second batch from a patient with the specific type of RP that's caused by having an extra, faulty copy of that rhodopsin gene.
And this slide just perfectly illustrates what they discovered. On the left, in the healthy cells, you can see that rhodopsin protein flowing exactly where it needs to go, out to those antennas that detect light. But on the right, in the patient's cells, something goes terribly wrong. The extra protein, it just gets stuck. It piles up inside the cell's main body, creating this massive, toxic traffic jam that eventually kills the cell.
And we're not talking about a small overload. When they actually measured the amount of rhodopsin protein, they found a staggering 16-fold increase in the patient's organoids compared to the healthy ones. 16 times more.
So what this means is that the organoid wasn't just a model. It was a living, breathing recreation of the disease itself. For the very first time, scientists could literally watch as the genetic flaw caused that protein traffic jam in real time. This gave them a clear, undeniable target for a potential treatment.
Which, of course, brings us to the next big question. If you can accurately model the disease in a dish, can you also test a cure in that same dish?
So the team took their diseased organoids, the ones with that toxic protein buildup, and they introduced a small molecule drug called PR3.
Now, this drug was specifically designed to go in and basically turn down the dial on the genetic pathway that was making way too much rhodopsin. And the results were just remarkable. As you can see right here in this chart, the untreated patient's cells, that first bar, show this sky-high expression of the rhodopsin gene. But then, after the treatment with PR3, look at that second bar. The expression level drops way down, bringing it so much closer to the normal level you see in the healthy cells.
This was a massive win. The drug didn't just cover up a symptom, it went straight to the root cause of the problem and addressed it directly. By fixing the defect at the molecular level, right there in the dish, they got this powerful proof of concept that this kind of therapy could actually work.
So, what does all this mean for the future of vision and really for medicine in general? Because the implications of this technology go so much further than just this one disease.
These organs in a dish are total game changers across the board. Scientists are using them to model countless other genetic diseases, to screen thousands of potential drugs quickly and safely, and even to study how viruses attack different human organs. They're an amazing tool for just understanding our own biology, and they might even become a source for cells we can use in future transplant therapies.
Ultimately, what we're talking about here is the dawn of truly personalized medicine. Just imagine a future where, instead of just trial and error, a doctor could test a dozen different drugs on your own personal organoids, like a biological avatar of you, to find the one that works best, with the fewest side effects, before you ever take a single pill. That's the future this tech is building.
This research has given us a whole new way to see, both literally and figuratively. And it leaves us with this one thrilling question. Now that we can model sight in a dish, what other complex parts of ourselves will we soon be able to understand, repair, and maybe even create?